Right now, inside your kitchen, there is very likely a living organism responsible for more human progress than almost any invention you could name. It is in your bread. It is in your beer. It is, at this very moment, being engineered in laboratories to manufacture life-saving medicine. And you have probably never given it a second thought. Yeast is so small that no human being even knew it existed for most of recorded history. And yet, it quietly rewrote the course of civilization while nobody was watching. It turned wandering tribes into settled societies. It built economies. It toppled and enriched empires. And it eventually became one of the most valuable tools in modern science. All of that, driven by a single-celled fungus that early humans could not see, could not explain, and yet somehow learned to worship. So how did something invisible to the naked eye come to shape the rise and the survival of the human species? That is the question this story is going to answer. Because the history of yeast is not really a story about baking or brewing at all. It is a story about accident, obsession, secrecy, science, war, and finally, biotechnology. It begins thousands of years before anyone understood what fermentation actually was. In a moment, nobody planned, and nobody could have predicted. A puddle of grain and water, left out too long, in the wrong place, at the wrong time, that started to bubble on its own. What looked like spoilage was actually the first recorded handshake between humanity and a microorganism that would go on to feed billions, intoxicate empires, and eventually help doctors produce insulin. We are going to trace that journey from the very beginning, from ancient bakers who treated their fermentation starters like sacred treasure, to the French microbiologist who proved that the process was not chemistry, but life itself, to the American entrepreneurs who packaged that living organism into a product you could buy in a grocery store. Along the way, we will separate the myths from the documented science, because for most of history, people did not understand what was happening inside their dough and their casks. So they invented explanations involving gods and spontaneous life springing from dead matter. The truth turns out to be stranger and far more interesting than the legend. This is the hidden history of the organism that built civilization, one rising loaf and one fermenting vat at a time. And by the end, you will never look at a slice of bread the same way again. Picture a riverside settlement in the region we now call Iraq, roughly 10,000 years ago, long before anyone had a word for fungus, let alone yeast. A bowl of crushed grain and water sits forgotten in the heat. Wild yeast spores, floating invisibly on the air the way pollen does, drift down and settle onto the porridge. Within hours, bubbles rise. The mixture swells, sours, and gives off a sharp, faintly alcoholic smell. Nobody planned this. Nobody understood it. But somebody was hungry enough or curious enough to taste it anyway, and what they found was not spoiled food, but something new entirely. Lighter, tangier, and strangely satisfying. That accident, repeated independently across river valleys in Mesopotamia and along the Nile in Egypt, is very likely how fermentation entered the human diet, not through invention, but through negligence rewarded by biology. Wild yeast, it turns out, is everywhere. It lives on grain husks, on fruit skins, in the dust of a granary floor, riding the air currents into any bowl of standing liquid that offers it sugar and thyme. Ancient peoples did not seed their dough with yeast on purpose, because they had no idea it existed. They simply left things out, and the yeast found them. Once early bakers and brewers noticed that certain containers, certain wooden troughs, certain clay pots, seemed to produce this magic more reliably than others, they started reusing them without washing, unknowingly preserving colonies of living yeast cells clinging to the porous surfaces. That single habit, born of superstition rather than science, is the ancestor of what modern bakers call a starter culture. The Egyptians took this further than almost anyone. Archaeological residue from bakeries in Amarna and Deir el-Medina shows deliberate, repeated fermentation practices dating back over 4,000 years. And Egyptian art depicts bread and beer production as parallel, almost sibling industries, both dependent on the same bubbling, foaming, unexplainable process. Because they could not see the organism responsible, they did the only thing ancient minds reliably did when confronted with an invisible force that transformed the world around them. They gave it a spiritual explanation. Egyptian religious texts and tomb inscriptions associate bread and beer with the gods directly, crediting figures like Osiris with teaching humanity how to brew, framing fermentation as a divine gift rather than a biological accident. Beer was so central to Egyptian identity that workers building the pyramids were partially paid in it. A ration of thick, nutritious, faintly fermented liquid bread that provided calories, hydration, and a mild buzz all at once. In Mesopotamia, the Sumerians went so far as to compose hymns to Ninkasi, the goddess of beer, describing the brewing process in poetic detail without ever once suspecting that the true agent of transformation was a microscopic fungus feeding on sugar and exhaling carbon dioxide and alcohol as waste. They were, in effect, worshipping yeast without knowing its name, building an entire theology around a process they could taste, smell, and feel bubbling under their fingers, but never see. Here is a question archaeologists have been quietly arguing over for decades. Did humans settle down to farm because they wanted bread, or because they wanted beer? It sounds like a joke, but the theory is serious enough to have a name, the beer before bread hypothesis, and it hinges entirely on yeast. Grain is heavy, slow to grow, and brutal to harvest by hand. For a nomadic forager, chasing herds and following ripening wild plants across open country, cultivating wheat and barley made little immediate sense. But fermented grain, whether baked into a dense sour loaf or brewed into a cloudy, mildly intoxicating gruel, offered something foragers could not get any other way: a reliable, storable, calorie-dense product that also happened to make people feel very good. That combination, nutrition plus pleasure plus shelf life, may have been enough to tip the balance from wandering to staying put. Once you are staying put to protect a fermenting vat, you are, whether you meant to or not, building the foundations of a permanent settlement. Grain surplus meant something new in human history, food that did not have to be eaten today. And where there is surplus, there is property. And where there is property, there is guarding it. Egyptian bakeries, and we know this from residue found in ceramic vessels more than 4,000 years old, were not casual, one-off operations. The residue shows deliberate, repeated fermentation practices, batch after batch, meaning someone in that household or temple complex was tending a living culture over time, feeding it, protecting it, keeping it alive between uses the way you might keep a fire burning. That someone was, in every practical sense, a yeast wrangler, centuries before the word yeast existed. Think about what that actually required. No refrigeration, no sealed containers, no understanding of contamination, temperature control, or biology of any kind. Just an unwashed clay pot, a scrap of the last batch's dough or dregs stirred into the next one, and an almost superstitious insistence on doing it exactly the same way every time, because the one time you did not. The magic sometimes failed to show up. Egyptian brewers and bakers functioned as parallel guilds, often working side by side, since the base mash for beer and the base dough for bread were nearly identical, differing mainly in water content and what happened to the mixture afterward. A starter culture, a living, working colony of wild yeast passed down through a household or workshop, was valuable enough to be treated like an heirloom. Lose it, and you were not just out of bread for the week. You were back to hoping the wind delivered a usable spore onto your grain mash by pure chance, praying to Osiris or Ninkasi and waiting. It is a strange thing to consider. The earliest form of intellectual property in human history may not have been a design, a song, or a recipe in any written sense at all. It may have been a smell, a texture, a living population of single-celled organisms nobody could see, passed hand to hand, pot to pot, generation to generation. Because whoever held on to it held on to the ability to reliably feed and intoxicate their community. Civilization, in other words, may have begun with someone refusing to throw out a dirty bowl. So which was it? A gift from the gods, or a smell nobody could explain? Ancient peoples were not stupid. They watched grain and water turn into something that bubbled, rose, and made people feel warm and loose-limbed, and they needed an explanation that fit the tools they had, which were not microscopes, but stories. The Egyptians settled on Osiris, a god generous enough to hand down the secret of brewing as a kind of technological gift to humanity. The Sumerians praised Ninkasi in hymn form, describing the process step by step, the soaking, the baking, the mixing, without ever once identifying what was actually doing the work. Other cultures leaned on an even older, vaguer idea that would echo for thousands of years afterward: spontaneous generation. The belief that life could simply emerge from dead or non-living matter given the right conditions. No divine intervention required, no invisible organism necessary, just decay somehow curdling into vitality. Mud-bred frogs, rotting meat-bred maggots, and soggy grain, left alone long enough, apparently bred bubbles and flavor, and the ability to make dough rise on its own. It was a tidy explanation for a world without instruments capable of seeing anything smaller than a gnat. Here is the problem. None of it was true, and the truth turns out to be almost stranger than the myth. There was no divine breath breathed into the dough. There was no life spontaneously assembling itself out of inert grain paste. What was actually happening, invisible to every single person involved for thousands of years, was colonization. Wild yeast, a naturally occurring airborne microorganism drifting through the atmosphere in staggering quantities, was landing on exposed grain and water mixtures and simply doing what yeast does: consuming sugars, exhaling carbon dioxide and alcohol as waste, and multiplying exponentially in the process. The bubbles were not divine breath. They were metabolic exhaust. The rising dough was not a miracle. It was gas production from a living population feeding itself. Every gift from the gods' moment in the ancient world was, biologically speaking, an uncontrolled, unintentional, airborne infection that happened to taste incredible. Think about that for a second. An entire mythology of grateful worship built around an event that was actually just a single-celled fungus, doing what single-celled fungi have done for hundreds of millions of years, completely indifferent to whether anyone thanked it. The ancients were not wrong to be amazed. They were simply looking in the wrong direction for the source of the miracle, up toward the heavens. when the actual answer was floating in the air around them the entire time, small enough to land on a piece of dust, patient enough to wait for the right bowl of grain paste to go unwashed. It would take thousands of years, a very particular kind of obsessive curiosity, and a piece of hand-ground glass for anyone to finally look the right way, and to look close enough to catch it in the act. For roughly 6,000 years after those first accidental fermentations in Mesopotamia and Egypt, nobody on the planet knew what yeast actually was. Not the Egyptian bakers guarding their starters like treasure, not the Sumerian priests singing hymns to Ninkasi, not the Greek and Roman winemakers who inherited and refined the process without ever once suspecting a living creature was doing the work. Fermentation was simply something that happened, a transformation as mysterious and unexamined as the changing of the seasons. It took a Dutch cloth merchant with no university training, no scientific credentials, and an obsessive amateur's hobby to finally drag the truth out of the shadows and into focus. His name was Anthony van Leeuwenhoek, and he ran a modest textile shop in Delft, in the Netherlands, in the second half of the 17th century. To inspect the quality of the fabric he sold, he needed to count threads with precision, and the magnifying lenses available at the time simply were not strong enough. So, he did something almost nobody else bothered to do. He started grinding and polishing his own glass lenses by hand obsessively, refining the curvature over and over until he had built simple microscopes more powerful than anything the official scientific institutions of Europe possessed. Then, because his curiosity did not stop at cloth, he pointed his homemade lenses at everything around him. Pond water, blood, plaque scraped from his own teeth, and, at some point, beer, what he saw down there, floating in the murky liquid, changed the trajectory of human food production forever. Tiny round and oval-shaped globules, distinct, countable, clearly organized structures rather than random specks of dust or debris. He was almost certainly looking at yeast cells, the very organism that had been silently transforming grain and grape into bread and beer since before written history began, and he was likely the first human being to ever lay eyes on it directly. He described these structures in his correspondence and observations, cataloguing them alongside the other microscopic life he was busy discovering, essentially inventing an entire invisible universe that no one had previously known existed. But here is the part where the story stalls out for nearly 2 centuries. Leeuwenhoek could see the cells. He could not prove what they were doing. He had no way to demonstrate that these tiny globules were alive, that they were feeding, growing, and multiplying, and that their activity was the actual mechanical cause of fermentation, rather than some incidental passenger floating along for the ride. For generations afterward, the scientific consensus remained stubbornly chemical. Fermentation was widely treated as a purely chemical reaction, a kind of decay or breakdown process with no living agent required, an idea that fit comfortably within the scientific frameworks of the time. The globules under the lens were treated as a curiosity, not a cause. The dough kept rising, the beer kept bubbling, and the true nature of the organism responsible remained frustratingly, maddeningly unconfirmed, waiting for someone with the tools and the audacity to prove that fermentation was not chemistry happening to yeast, but life being lived by it. That someone arrived in 1856, and he did not come from a laboratory chasing scientific glory. He came from a beet sugar distillery in the north of France, where the owners were losing money to a mystery they could not explain. Vats of fermenting liquid that should have turned to alcohol were instead turning sour, ruined, unsellable, and nobody could tell them why. The man they called in was a chemist named Louis Pasteur, then in his early 30s, already known for meticulous work on crystals and molecular structure, but about to pivot his entire career towards something far messier: spoiled beer, ruined wine, and rotting milk. Industrial France in the mid-19th century ran on fermentation the way it ran on coal and rail. And fermentation was failing constantly, unpredictably, and at enormous financial cost. Pasteur did what Leeuwenhoek 2 centuries earlier could not. He did not just look at the yeast, he tested it. Under his own microscope, Pasteur observed that healthy fermentations were dense, with the same small round globules Leeuwenhoek had sketched, while spoiled, soured batches were dominated by different, rod-shaped organisms entirely, contaminating bacteria that had outcompeted the yeast and produced acid instead of alcohol. This was the breakthrough. Pasteur demonstrated, through careful and repeatable experiment, that fermentation was not spontaneous, was not purely chemical, and was not incidental. It was biological. It was caused by living organisms consuming sugar and producing alcohol and gas as the direct byproduct of their own metabolism. And when the wrong organisms got into the mix, the whole batch turned. Living yeast made beer. Contaminating microbes made vinegar. The distillery owners had not been fighting bad luck. They had been fighting an invisible microbial turf war they did not know existed. Pasteur's solution was elegantly simple, and now bears his name. Heat the liquid gently, just enough to kill off unwanted contaminating organisms without ruining the flavor, then seal it away from new contamination. Pasteurization did not just save French breweries and wineries from spoilage. It effectively founded the science of microbiology as an applied, industrial discipline, transforming fermentation from folklore and guesswork into something that could be measured, controlled, and engineered. Governments and industries took notice immediately. If living organisms could spoil beer, they could spoil milk, meat, water, and wounds. Pasteur's work on fermentation became the foundation stone for germ theory itself, reshaping medicine, food safety, and industrial standards across the Western world within a single generation. Think about that for a second. The push to protect the profit margins of a handful of French brewers and vintners ended up rewriting the rules of human health. And at the center of it all, invisible, ancient, and finally understood, was the same organism that had been rising bread and bubbling beer for 10,000 years. Now identified, isolated, and about to be put to work at an industrial scale nobody in ancient Egypt or Sumer could have imagined. For 10,000 years, yeast had been a guarded secret, passed hand to hand in the form of starter cultures, sourdough sponges, and reused brewing vats, treated as a household treasure whose true nature nobody understood. Pasteur's discovery cracked that secrecy wide open. Once fermentation was understood as the work of a specific living organism, the obvious next question became commercial. If yeast was a living thing that could be identified, isolated, and cultivated in a laboratory, then it could also be manufactured, standardized, packaged, and sold. The age of the guarded starter culture was ending, and the age of yeast as an industrial commodity was about to begin. This transition did not happen overnight, but it happened fast by the standards of a 10,000-year-old tradition. Bakers who had once nursed a single starter culture for years, feeding it, protecting it, and passing it down like an heirloom, suddenly had the option to simply buy fresh yeast in a standardized, reliable form whenever they needed it. That single shift changed the economics of baking forever. No longer did a bakery's success depend on the health of one irreplaceable, temperamental colony of microorganisms nursed in a back room. Now it depended on supply, distribution, and brand. Entrepreneurs recognized the opportunity immediately. Commercial yeast production plants began isolating specific, reliable strains of yeast, cultivating them in bulk under controlled conditions, and pressing the resulting biomass into stable, shelf-ready blocks and cakes that could be shipped, stored, and sold like any other grocery item. This was fermentation without the folklore. No hymns to Ninkasi required, no ritual feeding of a starter passed down from a grandmother, just a product, uniform and dependable, manufactured to a specification. Bakeries that adopted commercial yeast gained a level of consistency that had simply been impossible before. Bread rose the same way every single time, on a predictable schedule with a predictable texture, because the organism doing the work was no longer a wild, semi-mysterious houseguest, but a domesticated, factory-farmed commodity. Household baking transformed as well. Recipes that once called for maintaining a live starter now called for a small cake or packet of commercial yeast, dissolved in warm water and used once. The relationship between the home cook and the organism changed fundamentally. Yeast went from being cultivated to being consumed. This was, in its own quiet way, as significant a shift as the original domestication of fermentation itself back in Mesopotamia and Egypt. What had once been a living inheritance, tended and protected because it could not simply be replaced, became a disposable ingredient, purchased fresh whenever needed, because the factory could always make more. The marketing followed the manufacturing. Companies built entire advertising campaigns around convincing households that their bread, their baking, their family dinner table depended on a specific branded yeast product rather than whatever wild culture happened to be living in the flour bin. Trust, once placed in a starter passed down through generations, was now placed in a label on a package. It is worth pausing on how strange this actually is. An organism that ancient peoples worshipped as divine, that medieval bakers protected like a family secret, had within a few decades become something you picked up at the grocer's alongside flour and salt. Manufactured at a scale and with a reliability that no single household starter could ever match. The invisible force that helped drive humanity towards settled agriculture had been fully tamed, fully industrialized, and fully commercialized. And once yeast became a mass-produced commodity, it was only a matter of time before governments and militaries realized it could serve purposes far beyond the bakery. Before we get to how yeast ended up on the front lines, let's pause for a second. If you're finding this deep dive into the microscopic organism running your bread and your beer even remotely interesting, do yourself a favor and hit that subscribe button. This channel exists to dig up the hidden histories behind the ordinary objects sitting in your kitchen right now. And yeast is one of the strangest ones we've covered. All right, back to the war effort. Because once yeast left the household and moved into the factory, it stopped being just a baking ingredient and became something governments actively rationed, promoted, and stockpiled. When wartime food shortages hit, flour and sugar were suddenly precious, and home baking was reframed by governments not as a domestic chore, but as a patriotic act. Posters and pamphlets urged citizens to bake their own bread rather than rely on commercial bakeries, to stretch rationed ingredients further, and to keep a reliable supply of yeast in the pantry as a form of food security. The packaged yeast companies that had spent decades building consumer trust suddenly found themselves cast as essential suppliers to the war effort, their branded cakes and blocks framed in advertising as tools of national resilience rather than mere convenience. But the more surprising twist is what happened to yeast outside the oven entirely. Nutritionists and military planners discovered that yeast itself, even without being used to leaven anything, was a remarkably dense source of protein, B vitamins, and minerals. Dried and deactivated so it would no longer ferment, yeast could be added directly to food supplies or consumed on its own as a supplement, delivering nutrition in a compact, shelf-stable form at a moment when fresh meat, dairy, and produce were often difficult to secure. Soldiers and civilians alike were issued or encouraged to eat yeast-based supplements to guard against deficiency diseases brought on by restricted wartime diets. This was yeast performing a function almost nobody who baked with it a century earlier would have predicted. Not as a rising agent, not as a fermenting agent, but as a nutritional stopgap, keeping bodies functioning when the food chain itself was under strain. Think about that for a second. The same microorganism that Sumerians once credited to a goddess, that Egyptian bakers guarded like treasure, that Pasteur finally proved was alive rather than magic, was now being handed out in tablet or powder form to keep hungry populations from getting sick during a global crisis. It's a strange kind of full-circle irony. Yeast began as something humans could not see, but could taste, then became something they worshipped, then something they industrialized and sold. And now it had become something they swallowed directly for survival, stripped of bread, beer, and ritual entirely, reduced to pure biological utility. The organism did not change. Its relationship to human civilization kept transforming instead, each era finding a new use for the same tireless invisible worker. And once science had learned to strip yeast down to its nutritional and biochemical essentials, it was only a short leap to start manipulating that biology directly, which is exactly where the story goes next. Out of the ration kit and into the genetics lab. By the second half of the 20th century, yeast had already been worshipped, guarded, bottled, and rationed. What happened next was stranger than anything the Sumerians could have imagined when they sang hymns to Ninkasi. Scientists discovered that yeast cells were not just useful, they were convenient. In a way almost no other organism on Earth could match. Yeast reproduces fast, its genetics are relatively simple to manipulate, and its cellular machinery for building proteins turns out to be remarkably similar to that of far more complex organisms, including humans. That similarity became the foundation of modern biotechnology. Researchers realized that if you inserted human genetic instructions into a yeast cell, the yeast would follow those instructions as if they were its own, faithfully manufacturing proteins it had no business producing in nature. Insulin, the hormone diabetics depend on to survive, had once been extracted painstakingly from the pancreases of slaughtered pigs and cattle. Genetically engineered yeast and bacteria offered a cleaner, more scalable alternative, turning fermentation vats into pharmaceutical factories. The same biological process that once turned barley into beer was now turning sugar into medicine. Call it fermentation's second act. Vaccines, enzymes, biofuels, and industrial chemicals all began flowing out of yeast cultures grown in steel tanks that looked more like refineries than breweries. The humble organism that Leeuwenhoek squinted at through a handmade lens had become one of the most heavily engineered life forms on the planet, a microscopic laborer conscripted into the service of modern medicine and industry. And yet, underneath all that engineering, it was still doing the same basic thing it had always done. consuming, converting, producing, multiplying, following the same metabolic instincts that once caused a bowl of forgotten grain mash to bubble on a Mesopotamian shelf. Which brings us back to where we started. Remember that number, that almost absurd scale of yeast's footprint across bread, beer, and biotechnology. It is not hyperbole. It is the direct, traceable result of 10,000 years of accident, ritual, guesswork, industrial ambition, wartime necessity, and laboratory precision, all stacked on top of a single-celled organism too small to see without help. Every loaf rising in an oven right now, every fermenting vat of beer, every vial of lab-grown insulin sitting in a refrigerator, all of it traces back to the same invisible lineage, the same restless microbe, doing the exact same thing it has always done, quietly and without credit. Civilizations rose, empires funded science to stop it from spoiling, entrepreneurs packaged it, governments rationed it, and biotech companies eventually rewired its genetic code. And through every single one of those transformations, yeast itself never changed. We did. Humanity spent 10,000 years chasing, worshipping, exploiting, and finally reprogramming an organism it could not even see, and it turns out the smallest thing in the room was running civilization the entire time.